In this video, I'm going to be discussing a recent project coordinated by Iridia and run as a proof of concept for the Global Alliance for Genomics and Health, or GA4GH. GA4GH have developed a Genomics Api to facilitate federated analysis on genomics data held in different locations and to allow the exchange of genomics information across multiple organizations. This API is implemented as a web service and can be plugged into different platforms or genomic analysis pipelines to allow different organizations to collaborate on a global scale by pulling in information from these different locations and allowing analysis to be run on them together. In this specific project, Iridia worked together with several participating sites who each set up the GA4GH API and loaded in a set of sample data for the project. This sample data was derived from the 1,000 genomes data by selecting a few of the samples from that project and distributing them across the different sites. The participating sites for the project were University of California, Santa Cruz, or UCSC, Stratified Medicine Scotland, or SMSIC in Glasgow, Scotland, Queen's University Belfast in Northern Ireland, EMC Research and Development in Moscow, Russia, and the Royal Melbourne Hospital and Biogrid in Melbourne, Australia. So each of these sites set up the GA4GH API and loaded in the sample data that Iridia provided for them. And you can find out more information about GA4GH, about the project, and how to set up their API at your own site here. They've got documentation and also the demonstration server with the 1,000 genomes data in it. Now this demonstration server is actually hosted by UCSC, so that's the API we use for that particular site. But the rest of the sites had to set up the server for this project and use the data that Iridia provided for them. So for this project, we used Iridia software, Analytics Agility. We set up a workspace, GA4GH Federation POC. This is the platform where we're going to bring together the data from the different sites and run our sample analysis pipeline. So within here, we can create R scripts. There's an R console which we can use from within the web platform, and this is what I'm going to be using to pull in data from the different servers and run some analysis on them. So first of all, before being able to do that, GA4GH have set up a Python client library, but within the platform we use R typically. So first of all, I've set up some functions for allowing our users to call this API in an easier way than just doing it all manually. So I've set up some functions that allow you to do that. So first of all, when we run the R console, these scripts are sourced, so we've got access to these functions in the R console. So I'm going to be running through this script here, poc.r, and I'm just going to run through it line by line or few lines by few lines to show how we set up different clients pointing to the different servers and then pull in information from each one and then finally run a little bit of analysis on them. So in this case, what I'm going to do is pull in some variant information from each of the sites, pull that together, and then the simple analysis I'm going to do is just work out the allele frequency for each of the each population at each individual site and compare these with the allele frequency in the overall population, which is the 1,000 genomes population. So first of all, these first few lines set up some clients to allow us to interact with the APIs at each of the different sites. These lines here just allow us to quickly go off and pull back some data sets from each of them. So running a simple query against each of the sites just to make sure that we can access each of them. So this has returned a list with five elements, and each element is from each participating site and contains information about the data sets at that site. Next, I'm going to just look at the API at SMSIC. First of all, I'm going to just have a look at the data sets there. So this is just one of those elements of that list that was just returned previously for SMS. This is the data set within the API that was set up for the proof of concept project. It's got an ID which is unique to this API, and we can use this to query things within that data set. And in this case, that's going to be some variant sets which contain the information Iridia provided to each of the participating sites. Each sample provided to the participating site is contained in a variant set. So running this query to search through all the variant sets within a particular data set returns a list of 10 elements with a name corresponding to the sample ID from 1,000 genomes. And again, it's got a unique ID unique to that this API, which we can then use to pull out variants from that variant set. So this is what this query is doing. And as each one of these is run, it's telling you exactly what's being sent. So this is just a JSON body which is being sent to this address. So that's returned, and it's pulled back all the variants from that variant set that we specified within chromosome 21. So far I've been looking at things in these list objects, which aren't as nice to look at as other objects in R. So this function here is just going to instead convert that into a data frame, which is R's usual tabular format for containing data, which makes it a bit easier to work with and a bit easier to view. So if you're familiar with VCF files, you'll see that this is essentially information that's would be contained in a VCF. So you've got the position and the reference and alternate basis of each variant, as well as an ID, because that was supplied within the VCF file as well. So that was just for one of the variant sets at SMS. Just running these two functions, these two lines here. This is going to loop through all of the variant sets within SMSIC and bring them all back, and then join them together into an overall data set containing all the variants from SMS. So there's 10 variant sets, so it's going to loop through, do this 10 times. Each time it's sending to a slightly different ID. That ID corresponds to the variant set ID within the API. Okay, and now that's run. I've just with this line here, I'm just displaying the first few rows from that from the results of that query. So we can again see that this is similar to before, but I've also added on this other column to tell us which variant set this variant comes from. So that gives us all the variants from SMSIC. So next I'm going to rerun this for each of the other participating sites, or each of the other participating sites that received sample data from Iridia. And this is going to do the same thing. It's going to run through, collect variants from each of the variant sets within that API, and join them together into one large data set. So once this is finished running, we'll have 4 sets of data, and each one contains all the variants from one of the participating sites. Okay, so now that's finished running. As it was going through there, it was printing out again the first few rows of each set of variants from each participating site. So just to show that it's returning a similar result set each time, just with some slightly different variants and from different samples. So next, we want to pull in some data from the API at UCSC, which contains most of the data from the 1,000 Genomes project. So what we're going to do here is we're going to pull in the variants again, but there's some extra stuff hosted at the UCSC API. So there's actually some annotation being done on the data in here. Some of this has been done using VEP, or the Variant Effect Predictor, by Ensemble. And this will tell us things like what the consequence of some of the variants are in this data set. So we can use this we can join this together with the data we pulled in from our other participating sites to enrich this data And we're also going to compare this, the global or the overall allele frequency within the 1,000 Genomes project, with the smaller populations at each of our participating sites. So first of all, we're just going to pull in the variants once again. We're just looking at one particular variant set in the UCSC API. And then we're going to look at a different variant set which contains some annotation information. So this sends a similar query but to a slightly different variant set. And once that's finished running, we can join this together with the variants pulled in from the other participating sites. So you can see that's finished running. So this next bit I'm going to do is using an R package called dplyr. This is just a useful library for aggregating data and running typical operations or operations you would typically want to do on data frames in R or in tabular data in general. And so here what we're doing is grouping by the ID in the variant sets, and then counting how many of the samples in that variant set have that ID. So this is finding the allele frequency for these smaller populations. So just have a quick look at one of them. This has the ID here and then the allele frequency here. So we've only got a population of 10. So for example, 1 in 10 of the population have this variant, but 6 in 10 have this variant, and so on. So we've done that for each of the participating sites. And then this query here joins them all together to find variants that are common across all the sites, and then compare the allele frequencies across the sites, but also compare them with the allele frequency for the 1,000 genomes population. And then also join together with the annotation data. So here we're just going to look at the consequence of that variant, and if there is any, the gene that variant affects. There we go. And just to have a quick look at what that looks like. So this has got the same columns as before with the position within the genome, the reference and alternate bases, but we've also got these allele frequencies as well. And finally, the consequence. So these are a bunch of intergenic variants, and if there is a gene then that variant affects, then that will be recorded in this column. So just an example of some things you might want to run to have a look at to derive something from this data. This is filtering to include only variants that are rare in the 1,000 genomes population, so with the allele frequency less than 0.1. So we can just have a look at this data and see that, well, for example, this variant here is rare in the 1,000 genomes population, but in our sample populations at each participating site, it's actually, well, all the samples have this variant at these sites. So that might be a variant of interest for your particular population. And the second one is just going to filter and return only the variants that have a gene annotated for them. So that's a total of 20 variants, which are either upstream or downstream gene variants for this gene here. And this is the ensemble gene ID. And finally, now that we've derived this data set, we can write it to the database within Analytics Agility to save for later or run further analysis on. So I'm just going to jump to that. I've written it to this table allele frequencies, which I can find here. We can preview the data and see that this is the same as what I was looking at previously with the allele frequencies and position in the genome. And just as a quick example of some more analysis you could do on this, you can create some very quick visualizations. And the heat map works quite well to compare the 1,000 genomes allele frequency with one of your allele frequencies at your particular sites. So you can create that chart And what this shows is that we have a fairly good correlation between the 1,000 genome allele frequency and the allele frequency for the population of the Australian site. But there are a few that are off-center. So ones above this diagonal line here have a higher allele frequency within the Australian site than they do within the global population, and vice versa for below that diagonal line. And we can run this for other columns or other frequencies within this data set. So that example was all done through the R console. I'm just going to show quickly how we can expose a user interface to these operations to make it a bit easier for users to get to grips with. So for example, this is using RShiny and creating a mini app in Analytics Agility, which allows other users to come in and interact with their data, or in this case, interact with these APIs. So here I can select my host site, and then I can create a query by using these different input boxes, and this brings back the data and presents it in a nice table here. And you can run through the same thing we were doing in the console, find all the variant sets within that data set. So that's these samples again, and then find all the variants, or a sample of the variants within that variant set. So this is, once again, detailing the genomic position of each of these variants. And once we've done all this analysis and pulled it together, you might want to visualize it in an interesting way, or present this back to someone in a digestible format. So this is another mini app within Analytics Agility, which creates a circles plot. And this is just an example of one way that you could present this information back once you pulled it all together and run your analysis. So, for example, in this track here, we've got the small variants or SNPs. So hovering over these points gives you some extra information, and these could be the variants we were looking at earlier. So just to summarize, what we've shown is that we can distribute data across several different sites. That's what we did by subsetting the 1,000 genomes data and distributing it across the different sites that Iridia were collaborating with for this project. We can then pull them together, pull the data from each of these sites, which is what we were doing in the R console. Join this together, which is what we did in a later point in the script. Join them together, find the variants that are common across these sites, and then derive some aggregates from them, or run other analysis. This was a very simple analysis, but it could be more complex, and we can also enrich that data with extra information from other sites that perhaps your site doesn't have access to, but another site does. So this is how different sites can collaborate and enrich their information by bringing it together. And then once you've done that, you can present this back in a nice digestible format to anyone else who wants to consume the outputs of your analysis.